Okay. Okay. Again, Assalamu alaikum, dear students. So the topic is mentioned on your textbook page number, that is 155, and this is included in your condensed or reduced syllabus. Um, so as you know that proteins are made in the cell. So uh, RNA is involved in making all this protein. The information is present in DNA, but the cell uses RNA to make protein. So, how a cell uses its DNA to direct the production of a particular protein, this question arises in your mind: where in the cell are proteins made? This is related to the both questions are same almost. So a question can come like these two questions to describe the protein synthesis in the cell. So we can answer these questions by pulse labeling. So first you must know what is pulse labeling. Placing cells for a short time in a medium containing radioactive amino acids, the cells will take up these radioactively labeled amino acids for the short time they are exposed to them. So once these radioactive labeled amino acids they enter into the cell, then our observation will be that we will see about we will see these cells, so where the radioactive proteins are appearing for the first time. So these experiments were performed in the past, and the scientists were amazed to see that the proteins were these amino acids were appearing in protein molecule in the cytoplasm, not in the nucleus but in the cytoplasm. So it was surprising for them that the DNA is the genetic material, but the process is taking place within the cytoplasm. So we will all discuss this in this topic, that how cell uses its RNA to make protein. As you know, there is a organelle present in the cytoplasm, which is called as organelle involved in protein synthesis. You know the name ribosome. So ribosomes are also called as protein factories. So these are little, Ibrahim, change the page. Okay, okay. So I will, I have put my lecture on recording. You can get the recording as well. And I will give the record of these papers to you people as well, so that you can make notes on your own, or you can study these notes and you can make your own ones, or you can just study directly from them, because I have made these papers like this, that you can find it easy to study them. So as you know that ribosomes are the protein factories. These are little polypeptide making making factories. So these whenever their chemical study was carried out, it was found that these ribosomes are chemically complex, as they are equally made up of protein and RNA, and over 50 different kinds of proteins are making these ribosomes, and the RNA is also found in them, and this RNA is called as ribosomal RNA. And the presence of RNA indicates in the ribosomes that RNA molecules play important role in polypeptide synthesis. As you know, proteins are made up of polypeptide chains. So recall your knowledge of first year, where you have studied that amino acids join together by peptide bonding to make a polypeptide chain, and proteins are variously structurally arranged to make various molecules. So this was my introduction on the use of the RNA in the cell to make proteins. So as you know, there are three main kinds of RNAs. One is ribosomal RNA, and one is transfer RNA, and the third one is messenger RNA. So we will study about these RNAs. First one is ribosomal RNA. As previously, few minutes back, I have told you that ribosomes are chemically composed of protein and RNA, and the RNA found in ribosome is called as ribosomal RNA. So, ribosome, as you know, they provide the site on which proteins are made. So, within the ribosome, in fact, this is the transfer RNA which is providing the site for the polypeptide to be assembled. So, a rough diagram of ribosome I have drawn, where you will see that the two particles are together. So, next is transfer RNA. इब्राहिम, ये कोई वो कर रहा है, इसको मिटाओ.
एनोटेशन कर रहा है ना कोई.
ये कौन एनोटेशन कर रहा है?
कमेंट करता हूँ.
ये पहले से कर दिया करो.
ये ऐसे नहीं होता है.
ओके बेटा, जिसको नहीं लेक्चर अटेंड करना होता वो ना आया करे. So, come to the second number of RNA, that is the transfer RNA. Transfer RNA is also found in the cytoplasm. It is free floating in the cytoplasm, and it is smaller as compared to the ribosomal RNA and messenger RNA. So, these transfer RNA are of various kinds, and in human cell you can find 40 kinds of transfer RNA. So each transfer RNA is specific for a specific kind of amino acid. Usually it is found that 20 kinds of amino acids are involved in making proteins, and for these 20 kinds of amino acids there are 20 kinds of transfer RNA, each specific for a specific amino acid to make various kinds of proteins. So this was about transfer RNA. So up till now you have studied about ribosomal RNA and transfer RNA. So in transfer RNA, you will study that I have given a separate heading of function, because short questions can also come. Write down the functions of ribosomal RNA and transfer RNA or messenger RNA, or ये भी आ सकता है question where these RNAs are found and where they are made. So of course, you know that all RNAs are made in the nucleus, and then through the nuclear pore from the nucleus they come in the cytoplasm, and so you will see a figure at the end of this page as well, where you will see a torch-like structure I have shown, and this is a transfer RNA. Transfer RNA are usually clover-shaped, जैसे फानूस होता है इस तरह से. So each, as I told you, that each is specific for a specific amino acid. For example, I have shown a dot as an amino acid. So at one end of this transfer RNA, amino acid is attached with a specific sequence of nucleotide at its one end, that is called as anticodon. Anticodon is a sequence of three ribonucleotides. So, for example, UAC. So UAC is the anticodon of codon AUG, which is present on messenger RNA. I will discuss this in the diagram. So you know the little information I am giving you, which I will use in detail in the description of the diagram. That on messenger RNA, the message is present again in the form of three nucleotides, which is making a genetic code. So a genetic code is made up of three nucleotides. For example, on mRNA, this is AUG. A stands for adenine, U stands for uracil, and G stands for guanine. This is called as codon on mRNA. Its anticodon is present on transfer RNA. So this occurs in complementary manner, and I have discussed this complementary whenever I was describing you the structure of DNA. So you know more about it. I will further discuss it in the diagram as well. So you know that during polypeptide synthesis, that is during protein synthesis, transfer RNA molecules play a role. This transports the amino acids present at their one end to the ribosome. The ribosome is the site where proteins are made. So role of transfer RNA is to transfer the amino acid to the ribosome for use in building the polypeptide chain. So you know, when two amino acids join together, they join by peptide bonding. So the function of the transfer RNA is to position each amino acid at a correct place on the elongating polypeptide chain. So you will see that this was a description about two kinds of RNAs. Now comes the turn of the third kind of RNA, that is the messenger RNA. And you will find that this messenger RNA is designated as mRNA. So, what kind of this messenger RNA molecule is? It is a long single stranded molecule, and it passes from the nucleus to the cytoplasm through the nuclear pore. So, function of messenger RNA is that during protein synthesis, during polypeptide synthesis, mRNA molecule brings information from the chromosome to the ribosomes to direct the assembly of amino acids into polypeptides. So amino acids are scattered in the cytoplasm, and the function of transfer RNA is to bring them to the site where ribosome is attached to the messenger RNA, and in this way the message is the message transcribed in the mRNA is translated into a protein molecule. So, from this lecture, the conclusion which we are drawing, that is, there are three molecules. One mRNA, the other is tRNA, the third one is rRNA. Plus, there are some ribosomal proteins as the synthesis is taking place where on the ribosomes. So, ribosomal proteins and the enzymes present in the ribosomes, they make up a system. This system is what is doing, it is doing two things. One is a reading of genetic message, and the other is producing a polypeptide chain that a particular message specifies. So it is a principal component or apparatus, that is the three kinds of RNA, the ribosomal proteins, the enzymes. These are the principal component or apparatus that a cell uses to translate its hereditary information present in the DNA, present in its DNA. So the information is in the form of a message written in the code specified by the sequence of nucleotide in the DNA. So that is, this whole apparatus is involved in making of protein. So DNA is the boss which is residing in the nucleus, and it is not coming directly in the cytoplasm. It is sending its three managers, that is messenger RNA, transfer RNA, and ribosomal RNA in the form of whole ribosome to the cytoplasm to do the work of DNA given to them. So this apparatus reads the particular message. So mechanism of reading and expressing the genes in organism is called as central dogma. So if a long question on of this type can come, define central dogma. So and describe it. What are its two steps? So your answer will be certainly like that. The mechanism of reading and expressing genes in an organism is called as central dogma. It has two steps. One is transcription and the other is translation. I will further describe these all topic in detail from the diagram. So an RNA copy of each active gene is made, and at ribosome, the RNA copy directs the sequential assembly of the chain of amino acids. So that is the chain of amino acids is called as polypeptide chain, that is making a protein. So proteins are made according to the sequence of RNA, and the sequence of RNA is in fact the sequence copied from DNA. So the mechanism of hereditary apparatus is same in both prokaryotic and eukaryotic cells. So there are some minor differences in detail in gene expression, but with a major difference that prokaryotes lack a definite nucleus, but eukaryotes have a definite nucleus. Some minor differences are there, but the basic mechanism of hereditary apparatus is same, that DNA gives its information to RNA, and then RNA accomplishes the function. So the three kinds of RNAs are there. Um,
ये कोई फेक बच्ची आई है, 1 मिनट। बोलो।
जी, नाम चेंज करके कोई आई हुई थी बच्ची, उसको ब्लॉक किया है। ओके, इब्राहिम बात सुने। डायग्राम
अच्छा।
I will describe all this what I have told you through a diagram. So नीचे करना।
माइक बंद क्या?
अच्छा,
कौन सी?
मैं निकाल लेती हूँ ये वाली डायग्राम। सही, it's हाँ. Okay, so I have shared a diagram on the page where you will see that the central dogma of biology is the topic written. This is the terminology for the protein synthesis through RNAs. So RNAs are involved in making of protein. So this process is termed as central dogma, and this is about protein synthesis. And you will come across in this topic that two steps are written. One is transcription and the second is translation. Transcription is within the nuclear membrane pointer. use करना है मैंने. Hmm. ठीक. Okay. Okay. So okay. अच्छा ठीक है. Thank you. Thank you. Okay, so students, you will see that this one is the nuclear membrane, and this one, the arrow is indicating the nuclear pore in the nuclear membrane. This all is the inside of the nucleus, and this is the cytoplasm where this second step. I'm running. Ibrahim, Ibrahim, इधर आओ, ये running out of time हो गया. This meeting will end in 10 minutes. Okay, okay, मैं finish कर दूँगी इसको 10 minutes में. Okay बेटा, हाँ बस मैं इसको 10 minutes में finish करूँगी. इसको जल्दी जल्दी figure को देखिएगा. So you will see that this is the DNA, the red color, double-stranded DNA, and this one is a enzyme, that is RNA polymerase, used to make an RNA. So the DNA splits and its nucleotides are exposed. Then there are extra ribonucleotides, the nucleotides which are making the RNA, these blue color, and then they are joined with the help of these various enzymatic actions, and these nucleotides, ribonucleotides, are joined together according to the instructions present in the DNA. I will describe this detail in the next lecture, inshallah. So this is a general overview of the central dogma. Then you will see that mRNA, when it is made, it is transferred to the cytoplasm, and this is all these are the nitrogenous bases attached to the sugar. So you know that mRNA is a single-stranded long molecule, and the three nucleotides are specifying one genetic code that is called as codon. And you will see that from the nuclear pores ribosomal RNA is also coming out, and this is making the ribosome, which are the protein factories. And this one you will see the transfer RNA, that is also made from the DNA. All three RNAs are made from DNA, and through nuclear pores they come in the cytoplasm. You will say this transfer RNA is smaller. These are the amino acids, and the transfer RNA has a genetic code of three nucleotides, which is called anticodon. So codon and anticodon are complementary to one another. We will discuss in detail in the next lecture. So don't you worry, I will describe it in detail. So you will see this green color structure is the ribosome. This is the larger part of the subunit of the ribosome, and this one is the smaller one. So the place where the ribosome is present, there you will see messenger RNA is present. So the place where ribosome attaches to the messenger RNA, the codons are exposed, and according to these codons, the anticodon brought by the transfer RNA, they come, and the three managers, that is the ribosomal RNA and the messenger RNA, the transfer RNA, these together start working to make the genetic code present in the mRNA to decode it. So that is the formation of RNAs from DNA is called transcription, and the formation of polypeptide chain, that is you will find these pink color various amino acid molecules which are arranged by peptide bonding according to the instructions present on mRNA. This step of decoding of the genetic code of DNA in mRNA into a polypeptide chain is called as translation. So ribosomes moves in increment of three nucleotides, and as it moves, the three codons are, the one codon is exposed, and the one codon is made up of three nucleotides, and that is the transfer RNA function is to bring amino acid from the cytoplasm, which is attached at its one end, and brought it at a correct site on the mRNA to translate the message that is in the form of a particular sequence of amino acids. So to make a polypeptide chain, and you know polypeptide chain makes protein. So you will find uh, this this is again a general introduction. Again, the DNA, which is a double-stranded, this from this DNA RNA is made, and this process is called transcription, which carried out with the help of various kinds of enzymes involved in transcription. And then this RNA that is translated into a sequence of amino acid with the help of tRNA and ribosomal RNA. This step is called as translation. So that is, the polypeptide chain is variously arranged to make various kinds of proteins. So this is again a general description to tell you that DNA that is making RNA, this is called as transcription, and making of protein from RNA, this is called as translation. So these are the two steps, transcription and translation. This is a cell boundary, cell wall, cell membrane, and then you will find this is cytoplasm, nucleus. चैट क्या है देखो. नहीं, अटेंडेंस का कोई मसला नहीं है बेटा. ये मैं रिकॉर्डिंग पे भी मैंने रखा हुआ है. जितने लोगों ने ज्वाइन कर लिया वो तो देख सकते हैं इस तरह से, और बाकी मैं रिकॉर्डिंग सेंड कर दूंगी, तो बाकी स्टूडेंट्स भी इसको जो है वो हासिल कर सकते हैं. जी, तो मैं बात कर रही थी इस डायग्राम की. आपको ये नज़र आएगा, न्यूक्लियस है, इसके डीएनए से आरएनए बना, आरएनए एमआरएनए की शक्ल में ये मौजूद है. ये ट्रांसफ़र आरएनएज़ हैं जो मैंने कहा था क्लोवर शेप्ड होते हैं, और ये एक छोटी सी स्ट्रक्चर आपको नज़र आ रही होगी जहाँ पे राइबोसोम मैसेंजर आरएनए के साथ अटैच होता है, वहीं के कॉर्डन्स एक्सपोज होते हैं, और ट्रांसफ़र आरएनए उसके रिस्पेक्टिव एंटी कॉर्डन्स वाले अमाइनो एसिड्स ला के उस जगह पे अटैच करते हैं, और इस तरह से पूरा मैसेज जो है वो डीकोड होता है. तो दो स्टेप्स हो गए, ट्रांसक्रिप्शन और ट्रांसलेशन. सो ये भी आपको एक अलग से मैं पिक्चर्स दिखा रही हूँ। डीएनए एमआरएनए बना और फिर प्रोटीन बने। ये ट्रांसक्रिप्शन ये ट्रांसलेशन दोनों स्टेप्स को मैं डिटेल में अलग से करवाऊँगी आपको नेक्स्ट लेक्चर में, और उसकी मैं आपको भेज दूँगी कि कब मैं ले रही हूँ। और यहाँ पे आप देखेंगे वही न्यूक्लियस है और ये साइटोप्लाज्म है। तो ट्रांसक्रिप्शन जो है वो न्यूक्लियस में होती है और ट्रांसलेशन साइटोप्लाज्म में होती है। और ये आपको नज़र आ रहे होंगे, ये अमाइनो एसिड्स को साइटोप्लाज्म से उठा के ट्रांसफ़र आरएनए लेते हैं और उस जगह पे रखते हैं जहाँ पे राइबोसोम एमआरएनए से अटैच होता है और मैसेज डिकोड होता चला जाता है। तो थैंक यू फॉर द स्टूडेंट्स फॉर टुडेज़ लेक्चर। चेट पे ले आना ज़रा। है कोई और कोई प्रॉब्लम है, कोई क्वेश्चन आपको करना? अभी थोड़े से 2-3 मिनट है मेरे पास, टाइम ओवर हो जाएगा। 40 मिनट्स की क्लास होती है। जब भी मैं आपको टाइम दूँ इसका मतलब है उसके टाइम से 40 मिनट्स तक आपका क्लास होगी, उसके बाद फिर क्लास ओवर हो जाती है। स्टूडेंट्स ने 52 स्टूडेंट्स ने ज्वाइन किया था। कहाँ है? थैंक यू। अच्छा जी, एमसीक्यू बेस्ड पेपर्स की अभी फाइनल कोई बात हुई नहीं है, सजेशंस हैं, मीटिंग्स होंगी, उसमें डिसाइड होगा। जब हमारे पास नोटिफिकेशंस आ जाएंगे तो उस वक्त हम आपको गाइड कर देंगे। अभी हमने आपको पढ़ाया ही ऐसे कि जिसमें आपकी अंडरस्टैंडिंग आए, और एमसीक्यूज़ जब ही आप कर सकते हो जब आपकी अंडरस्टैंडिंग होती है। हम आपको रट्टाफिकेशन तो करवा